Thank you, Professor Bob Hill. It's a pleasure to be here, to be speaking here tonight to the audience, and especially because this is the University of Adelaide, and it's been my home for the last 3 years, and I hope this will continue in the future. As Professor Hill was mentioning, I will be talking about the dawn of animal life. And I have to stress the point, animal life, because sometimes we confuse the early evolution of life with animals. As you'll see here today, they're quite separate in time. And that's what I'm going to do now. I'm going to start by trying to position ourselves in time. When we look at the usual geological timescale that presents the history of Earth, the 4.5 billion years of the history of the Earth, we see that there's a lot of detail in the last 65 million years. That's because that's when the mammals took off, diversified, and we are mammals, and therefore we stress that, and we put a lot of interest into knowing how our group developed. And there's less interest or less detail as we go back to 550 million years, and then almost nothing in general in the first 4 billion years. However, when we actually look at the real time lapse, we see that before the appearance of life on Earth, we have about 500 million years, and that's 1/9. Very soon after the oceans started to form on Earth, about 4 billion years ago, 3.8 to 4 billion years ago, there was probably the beginning of life. But after that, we had about 3.5 billion years of single-cell life on Earth. Very, very long time. As you can see here, 7/9 of the history of Earth are exclusively unicellular life. But what we're going to be talking about here today is this time, right here, when we move from single-cell life to the Ediacara biota, about 555 million years ago, and right after the Cambrian, the appearance of unequivocal multicellular life, and in the last 1/9 of the Earth's history, we have the appearance of animals. And one of its first representatives on Earth is the Emu Bay Shale here in Kangaroo Island, which is dated at about 512 million years ago. So in the Ediacaran, we're looking at these sort of organisms. We'll talk about them. And then in the Cambrian, we see things that we can already recognize. We see something that is probably capable of swimming. It's got eyes. It's got appendages. You can actually relate to it because it's not that different from what we see nowadays. So why did I come to Australia? Well, Australia, and in particular South Australia and Adelaide, is the only place in the world where you have, within a few hundred kilometers, the first elements of multicellular life, those Ediacaran fossils that I was talking about, and also the first animal fossils. Those two windows into what happened just before and just after the appearance of animals is why Adelaide is so important. It's unique. In Canada, they have representatives of both, but they separate by thousands of kilometers, 3,000 to 4,000 kilometers away from each other. There are some in Siberia, again, very remote, very inaccessible, and so on. So Australia, particularly Adelaide and the state, is the perfect place to research this. So we're going to be focusing on the Ediacaran of the Flinders Ranges and the Cambrian rocks in Kangaroo Island. So what is the Ediacaran? The Ediacaran period is a geological time period in between 635 and 542 million years ago. It has the first multicellular organisms, but the question is are they true metazoans? Are they true animals? We'll talk about that. And what is the evidence? Well, we have fossilized embryos. And again, there's a question mark because some plants have similar embryos. So the first cell divisions after conception. And they're found in Doshantou Formation in China of about 600 million years. That's what those fossil embryos look like. They're microscopic, about 0.5 millimeter in diameter. Then we have the Ediacara biota, the oldest macroscopic fossils of organisms, and those are about the oldest ones, 575. And then we've got the first animals with skeletons at the very end of the Cambrian, of the Ediacaran, sorry. And they look like this, Claudina. It's a very simple tube-in-tube sort of organism. So what are the first Ediacaran fossils that we know of? The oldest ones are not here in Australia. They're in Canada, and this is what they look like. This is the outcrop in the shore of Newfoundland. They're dated at about 565 million years, so they're older than ours, which are 555. And that's what they look like. They call rangiomorph. They are fractal growing organisms. They grow by addition and repetition of branching patterns, which is not how animals grow. So that's why these organisms, we don't really think they would have been animals, and we'll discuss what they could have been instead. They are, as you can see here, some of them are quite large, reaching 50 centimeters, 1 meter, and even longer. But they also go to very small and very highly detailed preservation. That's 5 millimeters of that very small front from Newfoundland in Canada. And these fossils were preserved in an environment that's very different to what we'll see later. These fossils were preserved in very deep water. This is the view, a reconstruction of what it would have looked like if we were able to go in a submersible to about 300 meters of depth, 200 meters of depth, and shine spotlight at the bottom of the seabed. We would have had fronds growing attached to incipient bacterial seafloors, and not a lot of diversity. 4, 5 taxa, 6, 7 at the most. This is the deep Ediacara biota, deep water Ediacara biota from Mistaken Point. So this is the time lapse we're looking at. We've got very few representatives. These are various groups, only a few, and the beginning of some others. But now we're going to go to Ediacara, and we here have a lot more diversity. We know from the Ediacaran fossils, from what's been studied already, that there's about 35 genera and 100 species. They are found in 30 different localities across most of the continents except Antarctica. And the ones that we're going to be focusing on now are these. These are the emblematic Ediacara fossils, which give the name from the Flinders Ranges to that geological period called the Ediacaran. So the latest, the only one actually defined in the last 120 years, geological period, is named after the hills west of the Flinders Ranges, the Ediacara Hills, which we work in. These are the sort of fossils. Here, the Ediacara biota is a lot more diverse than in the Newfoundland outcrops. We've got several, about 20 genera, and they look, some of them like these, Arborea, Sprigina, Tribrachidium, Dickinsonia. This is how they would look if we had the mother slab. If we had the perfect slab, this is what the slab would look like. It's got elements of all of those fossils that I'm mentioning. These are the real fossils. These are drawings of how they would look in the rock. And we try to envision that. How would it look, knowing the information that the environment is telling us? How would they look if we were able to go, like the other guys, in a submersible? These rocks are very shallow water. This is where they would have lived. They would have lived in about 10, 20, 30 meters of water, a bit more. They would have been living in intimate relationship with a microbial mat, a muck layer, with bacteria, cyanobacteria, and they would have had an intimate interaction with that. Some of them, as we'll see later, would have fed on it, others were attached to it, and there is evidence of small animals that would have been burrowing on that top layer. So something that I want you to share, that I want to share with you tonight, is that in the left-hand side here, you are seeing what will probably become the next state fossil emblem. It's still to be decided. We don't know which one of those it might be, but in a few months we will be able to announce, the minister or the premier will be able to announce that South Australia has a fossil emblem, and it will be one of the Ediacara biota. So what are we finding in the Flinders Ranges? Worked by my colleagues Jim Gelling and Mary Droser, and their teams have been working in the Flinders Ranges for at least 15 years, and they've been able to uncover complete beds. They turn them over, they look at the fossils, and each of those, that's like a jigsaw puzzle of all the slabs being turned over and put against each other. So we've got several square meters of beds. Here, each of those tags is a taxon, or a species. Each of those tags is an individual, and each of the colors is a different species. So we see that there is a large assortment, a large assemblage of organisms already in the 555 million year old seafloor. When we actually take into account several consecutive layers, our colleagues have estimated that there might be up to 3,000 individuals per cubic meter. So there was a lot of biomass already. Or multicellular organisms already were very, very common then. Not very diverse, as we said, about 30 genera, but very, very common. So what do we do with something like this? Our student, Felicity Kutz, has done her honors on this and is now developing her PhD on it. And she's put together this graph. Here on the left-hand side, we've got the frequency of number of specimens of the various groups, of the various organisms, animals, Dickinsonia, Kimberella, Papancrinus sprigina, and a number of others. And she's actually measured which orientation they're showing and how are they situated in relation to each other. So what we're seeing here seems to not be far from the same sort of line that we get when we study modern environments. So the ecology of Ediacara is already almost or very similar to what we see today. We'll also look at how these organisms were interacting, A, with each other and B, with the environment. Here we have how far they were reaching over that sediment that I was telling you about. They're all in intimate relationship with the sediment, with the water sediment in the connection. And we've got some of them are prostrate, they're lying on the surface. Some are standing up to more than 90 centimeters. So what that is telling us is that already back then, various organisms were trying to exploit the food, the elements they needed to grow in different ways. When I said, are these animals or are they not? Well, we've still got a lot of questions. One of the interesting proposals back in the 1990s was by Dolf Salacher, the late Dolf Salacher, who suggested that these group of organisms were not either plants, nor animals, nor fungi, which is the other multicellular group, but a new kingdom called the Bendobionta. This new kingdom would have grown by a kind of a matrix structure, and they would have had unipolar growth, bipolar, or radial growth. Some of these we have since realized they're probably more related to animals, but others are probably part of this large group that died out at the end of the Ediacara, never made it in competition with the animals. So which ones might actually be animals? So we've got here Dickinsonia, one of the most emblematic of them all. And if you look at that, you see those dashed lines. Take them off again. You see there's an imprint, very faint imprint. And what we're looking at is actually a Dickinsonia that is moving along the seafloor, digesting that microbial mat, and eventually being preserved and buried alive at its final resting position here. So it was digesting, we don't know how long, maybe a day, maybe a week, then moved on to digest another area, and so on, and eventually got buried and preserved. The other one that might probably be an animal is Kimberella here. It's been described as having the body plan of a mollusk. It's not unlike a mollusk. It's got a foot at the bottom. It's got an array of probably gas interchange or exchange surfaces, and it's got a proboscis that would have fed on that microbial mat. And how would it have done it? It would have scratched that surface, and it actually leaves those scratch marks, which are similar to the ones that are left by modern radula, by the radula of modern mollusks. The radula is the structure in the mouth that they use to scratch their food. So what happened after the Ediacara and biota? So we have the Cambrian explosion at about 540 million years ago, and for about 10 to 15 million years, we have what is called the evolution's Big Bang. What happened? Well, what happened is that we have a very sudden evolutionary radiation in very short time, and this brought about all the groups we know today of animal groups, animal taxa. So those were almost, not instantaneously, but they appear in the fossil record in a very fast manner. What else happened? We also have a radiation of what we call biomineralized organisms, things that have shells, exoskeletons, not bones yet, because vertebrates had not appeared in the fossil record, but we have all the other organisms, the spicules of sponges and so on. Those organisms appear. There was only one representative, Claudina, in the Ediacaran. Now they go berserk. They occupy a lot of niches. They grow in size, they grow in complexity, and so on. We also have the radiation of plankton, both phyto and zooplankton. We have the substrate revolution, and I'll show what that means in a slide later on. We have an increase in predation and complex interaction between organisms. And we have, the first complex reef systems. Those are the ones that are composing some of the first reefs that we find in the Flinders Ranges as well. The Flinders Ranges has got Ediacaran, and on top of that it's got the Cambrian. And in the Cambrian we see the limestones, and those limestones are made up by icoshyathes, which are these earliest reef builders, large organic reefs made up of sponges, calcareous hypercalcified sponges. This graph here might look overwhelming at first, but let me try to explain. Here on the horizontal, at the bottom, we've got time from the Cryogenian, the geological period before the Ediacaran, the Cambrian, Ordovician, Silurian, and so on, until now. And in blue, we have phyla. So there are maybe 1 or 2 phyla appearing in the Ediacaran, but then we have 85% of all known phyla in the fossil record appear for the first time in the Cambrian. I can understand. So what is a phylum? A phylum is things that are easy to recognize because they have a particular body plan, a particular structure in which they are built from a genetic point of view. Phyla, for instance, are the echinoderms, the sea urchins, the sea stars, the sea lilies, so on. Another phylum is the one that is the mollusca, that includes gastropods and cephalopods and so on. Another one is the annelids, bristleworms, earthworms, and leeches. And a very diverse one is the one of the arthropods, which includes things like crabs. What else appears in the Cambrian? Well, we have this other big hike in classes. 75% of all the classes appear in the Cambrian. So what is a class? That's the next hierarchical level under the phylum. And a class is Crustacea, for instance, the crabs, the chelicerates, the insects, and the myriapods. Those are the 4 classes, living classes within the arthropod. So that's the next level down, the next box within the arthropod box. This is what we call the agronomic revolution. What happened during the Ediacaran and transition to the Cambrian is that this algal mat, the organisms that lived in intimate relationship with it, and were buried on consecutive burial events, disappears, and you start getting organisms that are capable of plowing through it, digging into it, using it as an attachment. You lose that lamination, that is lost forever, and you start getting organisms that are capable of digging very deep into that sediment and swimming above in the water column. So this is the agronomic revolution I was telling you about. So what are the typical Cambrian fossils? Well, the trilobites that you see up here. We've got the echinoderms, the equivalent of modern sea lilies. We've got brachiopods, which are a bivalved organism, not a bivalve itself, because those are mollusks. This is a different phylum. We've got sponges. These are the typical quote-unquote Cambrian fossils. But with the types of research that we do here, that me and my colleagues do, is we're looking not only at the animals with shells, which actually constitute for the Cambrian about 27%, about a quarter of all the diversity, the generic or the species diversity of the time, is organisms with shells. But no, we're looking at what we're missing in normal Cambrian fossil localities. We're looking at the other 75%. And where do we look for that? We look in localities like Emu Bay Shale. In this case, this is Burgess Shale, but the same happens with Emu Bay Shale. We have the whole components of the fauna. We've got the soft-bodied organisms. Not only the soft-bodied organisms, but also the soft parts that they had in their bodies, from their muscles to their brain tissues to their gut system with their last meal, so on and so forth. This is what Emu Bay Shale is providing us about the earliest evolutionary event on Earth, which is the first for animals, which is the radiation of animals. This is Burgess Shale, this is Emu Bay Shale. This is what we study. Where are there localities like this? Well, the oldest one is Chengyang in China. We've got Emu Bay Shale, slightly younger here. Then we've got Burgess Shale. We've got a number of others. And as you can see, they're all around the equator. That's not very difficult to understand. It's similar to nowadays, where the biggest diversity, as we know, is in the reef areas, in marine environments, and in the rainforest in continental environments. My work, as mentioned before by Bob, started in the Burgess Shale. Burgess Shale is a locality up in the Rockies. These excavations I did 95, 97, and 2000. There are 2 months of excavation up in the middle of the national park, so we have to take all our equipment. This is how we do it. We would hire helicopters. They would take us up to the hill, and this is the bottom of the hill where we would camp. At July, the equivalent of November, December here. December, actually, beginning of December. And, of course, we would have to wait, in some cases, for that snow to melt to access the quarry itself. So where is it? This is Burgess Shale. This is Fossil Ridge, where Walcott, for those of you who might have read Wonderful Life by Stephen Jay Gould, this is where it all happened. Up in the middle, that's where Burgess Shale is. Mount Wapta, Mount Field, and you've got the Trans-Canada Highway and the town of Field down here. These are the 3 fossil-producing levels, called Walcott Quarry, the original one, 1909 to 1920. In the mid-1920s, Raymond from Harvard University, and then Collins Quarry, the Royal Ontario Museum, slightly later on, in the 70s and through the year 2000. This is at the end of the summer, when the snow had all melted. We were accessing what little rock Walcott had left behind, because all of the other he had collected, and it was down in Washington, not in Canada. Remember, this is Canada, but back then this wasn't a national park, and all the fossils discovered by Walcott and by Raymond were taken to the US. So the Canadians wanted to have their own collection, and that's what the Royal Ontario Museum did. So how do we study these fossils? This is Marella. It's one of the most common arthropods from Burgess Shale. That's the fossil with the aid of a microscope, very simple, with a camera lucida attachment, which is a mirror and a magnifying lens. We're able to trace with a lot of detail over each of the structures that we see in the fossil. Not only that, we actually try to understand each bit. What is each part that we're seeing in that mismatch of legs and hairs and so on? So that's what we do with each of the fossils. We produce a camera lucida, and with enough information, we're capable of producing a dorsal reconstruction of the organism in this case. And with a dorsal, lateral, frontal reconstruction, we can produce this. This is what we published in 2006 after we reviewed Marella. We revised it. And now I'm working with colleagues from Denmark in producing a reconstruction for a museum up there. So this is the internal anatomy, the blood system, and the gut, and the rest. So technology is allowing us to go that step further in trying to understand what these organisms look like. How were they made in the inside? Another information source is the rock itself. When you use polarized filters, this is the same specimen. This one up here is dry from really low angle light. The bottom one is underwater with polarized filters. You can see those 4 eyes were invisible in the original specimen. Unless you actually know what you're doing and you're playing with the light, you're putting water over it and so on, you don't have all the information. And that's what we're doing. We're extracting as much information as we can. The same happened with Anomalocaris. Anomalocaris is one of the better known, the most emblematic Cambrian fossil. It was originally described on this. Anomalo means strange, weird. Caris means crab, crustacean. And it looked like the tail, that thing there, looked like the tail of some sort of lobster, but they never found the head. And those legs, quote unquote, those things coming off each of those segments, were stiff. They weren't jointed like modern arthropods. So that's why they called it Anomalocaris. That's actually the first Burgess Shale fossil described. It was even before the Burgess Shale itself was discovered. It's quite interesting. What other things were found together in this same outcrop? Well, this thing here, originally called Peitoia, and it wasn't until we actually found the complete specimen of Anomalocaris that we were able to produce a complete reconstruction. And here, I've been able to bring a model of what Anomalocaris would have looked like. And this is a baby one, okay? We've got the eyes here, the two eyes. We've got the appendages here and here. We've got the lateral flaps on the sides, and we've got the flukes at the back, right? And the mouth would have been in the center right there, which is this thing here. So sometimes paleontology is building over what, most of the time, it's building over what other people have already done. We're not discovering, sometimes we're not discovering anything new. We're getting more details about things that were already there, that some person 100 years ago, 50 years ago had already mentioned or described. So with enough information, you can actually put together a mural like this one by Marianne Collins on what the Raymond Quarry has. The major predator is Anomalocaris. There's a number of other arthropods, sponges, brachiopods, mollusks, etc. Now let's come to Emu Bay Shale. This is where we are right now, and this is Kangaroo Island, as you all know. In the north coast, we've got Emu Bay Shale. Kingscote, Emu Bay is right here, across the Bay of Shoals and North Cape. The original fossils, only shelly fossils, not the ones with soft parts, were found in the western side of Emu Bay. That's why Emu Bay Shale is called Emu Bay Shale. It's in Emu Bay. But the quarry where we work and where the soft-bodied fossils were found is over here in Big Gully. So let's look at that in detail. The original fossils were found in the wave cap platform and the cliff. Unfortunately, the cliff is not stable enough, very risky, and the wave cap platform, the problem is you can only work for maybe 1 or 2 hours a day if there's a good tide. So what my colleagues did back in 2006 is put in an ARC application to try and dig the fossils further inland. We know that the beds come into here, they're the same beds, and they saw that there were some soft part preservation there. And they say, okay, let's give it a try. And so we opened what we now call Buck Quarry, named after the Bucks, which are the owners of the land. This is back in 2007 in September. That's me. That's our colleagues. I'll talk about them later. And this is a line perpendicular to the beds. You can see that the beds are tilting towards the north. We opened a pit, and then when we got to the best levels, we sprayed sideways. September 2011. This is looking north towards the mouth of the gully. On the right-hand side would have been the original locality. We are taking samples for geochemical analysis as well. We've taken 1.5 meters of a column to do the fine geochemical analysis to understand what the sedimentology, what the rocks are telling us about the environment in which those fossils were deposited, and also what might have affected those fossils, the process from the time they were buried, alive or dead, to the time we recover them. And this is what it looked like in our last excavation in September of last year. We will be going there in about 2 months' time again. So what are the typical Emu Bay Shale fossils, the ones we already knew about before we got this new excavation going? So Redlickia, the trilobites, we already mentioned them. They have a really hard exoskeleton. They are mineralized, just like crayfish, for instance, nowadays. So that part, they molt and they shed that skin, and so those parts are very common to be found. One of the most common components of Cambrian assemblages, of the fossils that are found in Cambrian rocks. But there were also soft-bodied fossils, as I mentioned. So Rhonescolex, a worm. We've got Tuzoia, an arthropod. It's got 2 eyes here. This one's been buried like a butterfly. Isoxes, it's got eyes as well. Myoscolex, which we're looking into because it's had a very tempestuous history of research. It's been placed almost everywhere. But since then, we've found and described a lot of new species. Among the arthropods and close allies, we've got Isoxes, Oestocercus, some Arthropodans, and a spiny Lobopodian. That's a velvet worm, marine velvet worm from the Cambrian. And we have had the chance and the honor of being able to name some of our species after people that are very important to our research. In this case, we've named this one after the Buck family, the owners of the land, and this one after one of our colleagues, Mike Gemmell, who's a very, very good fossil finder. So this is the sort of work, this is the end of the description of the fossils. Then we try and build over that, what were the relationships between those fossils. So you see these 2 pictures. They're quite different but quite similar. And there's only more than 500 million years of time between one and the other. The reason for this to be preserved is why we have the research that we have here on Emu Bay Shale. This is unique. There's nowhere else in the world where you have that preservation in a fossilized eye of 500 million years of age. So this is the paper where we described it, that we have a number of specimens. That's a robot-fly like the other one, a modern fly in comparison with these organisms. And what we saw is that these eyes had a lot of lenses. We were able to extrapolate, and we found that it had approximately 3,000 lenses. A lens is, in a compound eye of an arthropod, is the equivalent of a pixel in a camera. So the more pixels, the better resolution you have in your photograph. Same with this. The more lenses, or materia, the better resolution you're going to have with your eyes. So we saw that it had 3,000 lenses, which is way above. Those dots there are the trilobites of the time. The other information we have of lenses in the Cambrian and Ordovician is from trilobites, those mineralized arthropods that I was talking to you about. And it took about 60 million years for trilobites to get as many lenses. But we see from the soft part that that complexity was already reached right at the beginning of Cambrian animal life. The same happens with another aspect. We also looked at the size, and we see that the size of those ommatidia, those lenses, is very, very large compared to what other Cambrian trilobites had. Again, it took about 70 million years for trilobites to have such large lenses. These are located in the center of the eye, and that normally goes hand in hand with the bigger the lens, like the more open the diaphragm in a camera, the more light that goes into the eye, the better capacity to see in very dim environments. So this predator, again, the more acute the eye, probably the more of a predator you are. There tends to be a relationship between predation and capacity to see the prey earlier than the prey sees you. We see that these organisms were already right at the top of it. So what do we see if we were able to look through the eye of one of those trilobites with about, in this case, 84 ommatidia? That thing coming at it would have looked like this. There's something there. We don't know whether it's color or black and white, but we know there's something coming, there's movement. Modern-day horseshoe crabs have about 1,000 lenses, and that's how they would have seen this predator coming at them. However, the Emu Bay shale eye with 3,000 ommatidia clearly recognized what was coming at it. And now, after having seen this, you would also recognize this is a bad guy. This is Anomalocaris. And when I said before this is a baby, it's because we find these appendages which are keep a ratio of size with the rest of the body, and sometimes we find these appendages as long as that. So this organism, this animal, would have probably reached 1.5 meters in length in the largest specimens. So this guy was a ferocious predator, and it would have helped this other organism it would have been seen by this other organism quite clearly. And with the most developed modern arthropod eye, which is that of the dragonfly, in terms of lenses, somebody might point out mantis shrimps have more complex eyes. I'm not going to go in there, but in terms of number of ommatidia, that's how a dragonfly would have seen Anomalocaris. We know what the owners of those eyes are, except this guy. We don't know who is the owner of that eye. And some might say, wait a minute, if it's large, if it's got such acute vision, why is it not Anomalocaris? Well, I'll tell you why. Eyes in the Burgess Shale and Shenyang Anomalocaris look like this. They're large, they're in this case smooth. Let's look at the detail. We have here and here, there and there. We know what they look like. We know the shape, we know the size, but Burgess Shale and Shenyang, like this one here, that's Anomalocaris sarin, the Anomalocaris from Shenyang, from China, slightly older, and this is a detail. We know what the eyes look like, what shape they have, but they don't preserve the lenses, so we don't know what to expect until we actually came across this. Eventually, we did find the eyes of Anomalocaris, These are much larger than the other ones. And there's a picture of them here. We've got 3 specimens now. This pair of eyes, they've got the shape just like the Anomalocaris of Shenyang and China. They have a visual surface, this area in grey, which is covered in straight lines of hexagonal, very finely packed lenses. That is the eye of Anomalocaris. It's got many more lenses. Many? How many? Okay, well, we did the job. We worked out how many lenses there were in the y-axis, how many in the x-axis, and we counted about 16,000 on one side. Of course, you have to understand that Anomalocaris has lenses all around the eye. When this animal dies and drops to the bottom and gets compressed, you're only going to get one of the sides of the fossil. You're only looking at 1/2 of the visual surface of the eye. And that's 1/2 is 16,000, so two halves would probably have around 30,000 lenses. That's a conservative estimate. And that is more than the modern-day dragonfly. So the eyes of Anomalocaris were more acute than the most acute of modern-day arthropods after 515 million years of evolution. Very interesting. So what are the things we've been finding in Emu Bay that are interesting to, besides those single specimens or those arthropods? These organisms, the Vetulicolians, had been known for at least 20 years. They look like a big sack at the front. It's got a groove on the side, and it's got a segmented, quote-unquote, tail. And that's why, because of that segmented tail, which very much resembles that of an arthropod, they were thought to be originally arthropods. But then they found that there are structures in that lateral groove, that are used for filtering. And those structures are very much like the ones in deuterostomes, which includes us, vertebrates, and chordates by extension, but also echinoderms and hemichordates. So deuterostomes tend to have that sort of structure. There's a number of other structures inside that part that also put it in that tree. However, we didn't know where it fell within that. There were still some people, oh, no, it's an arthropod. That tail is unequivocally arthropod. Okay, so a few years back, we actually found Vetulicolians in Emu Bay. And this is what they look like. This is the holotype. In this case, this is the camera lucida. You know how camera lucida is produced now. And in this side here, what we have is the tail that's been folded backwards. That's why it looks a bit weird. Actually, there's a fine sediment in between the anterior part and the tail. So that means it's in a different level. It's been tumbled in the sediment and buried very much contorted. So anyway, this is the reconstruction. We were capable of putting those fossils together, finding a complete one, and being able to see, okay, well, our Vetulicolian, the Australian Vetulicolian, is very much like the Chinese one. It's got a big head, a big anterior portion. It's got the groove on the side. It's that line there. And it's got a tail, quote unquote, with segments, 7 segments, just as many as the Chinese one. And said, okay, so we do have a Vetulicolian. Let's see what else is in there. When we look at the Chinese Vetulicolians, we saw that the gut in the middle of the tail it varies in width, like almost every gut. It's quite wavy. It could be thicker and thinner depending on how much sediment or digestive material was inside. And in this case, we even have a little bit of poo pushed out behind or after the extension of the tail. That is obviously telling us we're looking at a gut with sediment infill, and when the animal was compressed, it spilled out a bit of the internal sediment into the surrounding matrix. However, what we found in our fossils was totally different. We had something with very straight lines, you can see here, and it didn't rip like a tube with sediment inside, which would have ripped and contents would have been spilled everywhere. Here, it was breaking in blocks. You can see that block there? See the blocks? See the block? And we went through the literature and said, okay, what sort of structure could it be that breaks in blocks? And this is a notochord. This is the notochord of a modern early vertebrate. The notochord is the part of the body that turns into the backbone. Our backbone is actually made up of blocks of bone, with cartilage in between the discs, and that cartilage is like the ones we're looking at here. Right? So this structure was not the gut that we're seeing in the other Vetulicolians. It's a different structure. After studying some of the other characters, we were capable of analyzing where would this group of organisms live, the Vetulicolians, where would they fit into the tree of life. These are the cephalochordates, those are the lancets, that's the vertebrates, the group that includes us, and oh, excuse me, this has gone crazy. Okay. Gee, oh, come on. Well, let me stop. We are here. That's where we are. Let's see if it doesn't go crazy again. Well, it does. That's not nice. Okay, let me just stop this. Escape. And I'll do it by hand if you don't mind. Okay, so we see that here we have the vertebrates. That's a group that includes us. And here we've got the group that has, among them, the tunicates, which is our closest group of chordates. And it's got things like, oh, of course I wouldn't have a pointer. It's got the, I'll use this. It's got a tadpole larvae and what is normally called, known as conjuvoids, or ascidians. So we know that Vetulicolians, based on the information we were capable of finding in the Australian fossils, we were finally putting them in the position in the tree of life. So what does the EBS fauna have? Well, we've got representatives of sponges. In this case, it's a picture of sponge from North America, but we have really good sponges. We've got early mollusks, two types of early mollusks. We've got the priapulids, those worms that I was telling you about before. We've got good examples of polychaetes, bristle worms. We've got the brachiopods that were very, very common at the time. We've got early arthropods of the likes of what we call Telsongai up there. We've got obviously Anomalocaris, as you've seen before. We've got representatives of the trilobites. We've got good tunicates, or sister taxa to tunicates. We've got some echinoderms that we've been finding just below the Emu Bay levels in the last two digs. And we've got the armored lobopodians, those marine velvet worms. So these work, and especially the work done by my colleague Jim Gelling, brought a number of years ago Sir David Attenborough to South Australia. Unfortunately, BBC is quite tight in its schedule, and he couldn't come to Adelaide and visit the museum, but we were able to invite him, and he came over and paid us a visit, and he saw both the collection of Ediacaran fossils as well as some of the fossils, obviously the eyes, which he knew about, and also some fossils with our colleague John Long, who named a Martopisces, the oldest fossil fish from Gogo, with an embryo inside after him, Martopisces attenboroughi. Not only people of the stature and the capacity of Sir David Attenborough have been attracted to what the Cambrian and Ediacaran is telling us about evolution. Also, these guys. You might have encountered them. They're about this size. And these are cards that Woolworths, good old Woolies, has put out there. So Marella here, now you know a lot about Marella. And Wiwaxia, two of the most emblematic Burgess shell fossils, have been shared with the kids. So it's good that the kids are now hearing about these things. They had an Anomalocaris, but it was so bad, I just didn't want to have it on screen. So what else is coming from Burgess shale type localities, from Emu Bay shale type localities? Well, this is a finding from Chengyang. Here they have, our colleagues have found a specimen that preserves the brain and the connection with the eyes. I'm going to do it this way. I'm going to use the pointer. So we've got the brain here, and we've got the nervous system connecting it, the optic lobes connecting it to the eyes and to the antenna in there and there. Not only the brain, which you might say, how could it be preserved, but also the circulatory system. As you saw from Marella, that was already known, but here it has a much more developed circulatory system, and this is what we have. We've got the gut in green, blue-green, which is well known in Cambrian arthropods. It tends to fossilize quite well. We have the brain from this specimen, and they put over that the circulatory system, which is this structure here, with a lot of veins, very similar to what we found in Marella. So just to take home a couple of messages. One of them has to do with how we interpret evolution based on what Cambrian fossils and Ediacaran fossils are telling us. One of the ideas is that evolution is an increasing complexity. As you saw from the eye, with visual systems, some of the most complex visual systems, I'm not saying human eyes, but some of the most complex systems of vision in other groups were attained very early in evolution. And one of the other ideas is that those groups apparently died out, leaving any descendants. Anomalocaris, no descendants. Wiwaxia, lost forever. Odontogryphus, no. Same with Deinonychus. So what happened is that during the Cambrian, some of those fossils, which at the time you wouldn't have been able to say, oh, this guy's going to make it, that guy's going to die, they were everywhere. Anomalocaris, they were everywhere. They were the apex predator. And we couldn't at the time have foreseen what was going to happen. However, they died out. So on a redefinition of Darwin's suggestion that there's a cone of increasing diversity, where you have in the Cambrian and Ediacaran very simple forms, and as time goes along, these forms just get more and more complex, and there's more and more branches. I have to agree with Stephen Jay Gould, what he proposed in Wonderful Life, that actually we're looking at a slightly different pattern. There is an increase in diversity, a diversification, because that's how species evolved, but the actual shape of that is different. It's not a cone, it's more like a tapestry. So if we were to be looking at the Cambrian, we would see, wow, there's a lot of branches, things that are even, that are now totally extinct, that have left no descendants. But when there comes a time of crisis, there's an extinction, and only a few groups make it across that. And those are the ones that re-diversify. So it's a slight change of understanding of how evolution progresses, or the evolution just moves along. The other idea that I want to put forward is another one that was suggested in its time by Stephen Jay Gould. This is how most people understand human evolution. Human evolution is a linear process by which animals, eventually humans, are getting more and more complex, more and more intelligent, more and more straight. However, when we actually look at the fossil record, that's not the case. This is what happens. We've got a bush, and the actual human history, it's very windy. It's not a straight line of getting more and more straight and getting more and more complex and more intelligent. This is probably one of the take-home messages. And I wanted to end by saying all of this research, like in most of the cases, is not a personal thing. This is about group effort. This is about sharing responsibility, sharing knowledge, sharing ideas, comparing, and coming up with explanations that might not have occurred to any single one of us. Here we have people from the University of Adelaide, myself, we've got the South Australian Museum, Jim Gelling and Mike Lee, we've got UniSA, Jim Jager, we've got UNE, our colleague John Patterson, we've got Natural History Museum in London, Greg Edgecombe, and our helpers like Mike Gemmell, our volunteers, Mike Gemmell, Rhonda Atkinson, and Marianne Binney, and Natalie Schroeder, and so on. This is team effort. You can get a lot further with team effort than you can by yourself, unless it's probably physics and you're Stephen Hawking. And of course, I have to acknowledge financial support from the ARC, the University of Adelaide, South Australian Museum, the Environment Institute, National Geographic, and the Spanish government of, the Spanish Ministry of Economy. Thank you very much.
Okay, thank you. We have time for a few questions. There are a couple of microphones, I think, circulating. So if anyone has a question, can you put your hand up and we'll get you a... There's one up the back.
I've brought a pen, so if we need to, discuss something, we've got a whole whiteboard to work on. We could be here until midnight.
We've got 5 minutes.
Just firstly, I wanted to say thanks for a fantastic presentation.
Thank you.
I love your enthusiasm about your field. Just one thing though, I know that what you're doing is very important with regards to life that followed on from here, but how do you make this sort of stuff?
Sorry, I missed that.
How do you make this sort of stuff sexy to the kids? Like you show them a Tyrannosaurus Rex or a Triceratops lose their minds.
Yep.
How do you present this sort of stuff to them?
Well, we have to play with what we have. The earliest animals don't look like anything that we see nowadays. First, second, don't have teeth, big, sharp, nasty teeth, like great megalodon, for instance, or a T-Rex. So we have to first realize that kids are interested in knowledge in general. If it's got that spice, oh my God, this could have eaten me, then it's got that extra interest. But otherwise, I think we have to be clear in saying science is something that goes along. As it goes along, it adds interest to it in form of new knowledge. The eyes, for instance, of Anomalocaris, the appendages, what we find I haven't even gone in there, but we find trilobite bits and pieces in the guts of these animals. We find fossilized poo with trilobite bits and belonging to Anomalocaris here in Kangaroo Island, which are not common elsewhere in the world. So you have to tell them what is out there. And most of them, they might need the rush of a T-Rex ripping a head off. I think we should be able to tell them that there's a lot more besides that. You can put it in an accessible way, and they will probably follow. I've had a chance to talk to scouts, and they would follow this talk for 40 minutes, and they were fine. So yes, it is difficult, but yes, it is really worth trying. It's quite rewarding.
Thank you also for a wonderful talk. Do you have any ideas what the crisis might have been that wiped out those different
groups? Yes.
Thanks.
Good question. Well, I haven't even had chance to do that, but I'm also working in the Ordovician, which is the next geological period after the Cambrian, and what we see is that Anomalocaris did not make it until today, but they were even bigger in the Ordovician. They were 2 meters plus, and they were occupying all sorts of ecological niches. They were filter feeders, like the equivalent of modern-day whales. They were huge predators, raking the seafloor and getting soft worms and mollusks to predate upon. So some of these groups lived on. Marella, there's a younger version of Marella, more spiny, in the Ordovician as well, of Morocco. That's where I was when they're in October. It's amazing. So we just have to keep looking for these Lagerstätten, these localities that have soft part information. That's opening up huge windows. And groups that we thought disappeared in the Cambrian, there's a major extinction, mostly with trilobites, as we know from the record. Trilobites go through a huge extinction at the middle of the Cambrian and at the end of the Cambrian. But some of these groups lived on. The soft ones we thought were gone. But no, when you actually open those windows, you find them. Some of those are still there.
Okay. Oh, we've got... Oh, sorry, another one? Yep.
Congratulations. Have there been any attempts to amplify DNA from any of the fossils?
Thank you. Thank you for that question. Well, if Alan Cooper was here, he would probably tell you that, unfortunately, we're way back in time. There's almost no organic, maybe a little bit of cuticle left, some biomarkers, but DNA doesn't last long. We estimate that unless it's very controlled conditions, like in a deep cave or frozen in the permafrost in the Arctic, DNA is lost within weeks or months. So unfortunately, no, there is no DNA, there is no protein, there's no polysaccharides. There's a lot of organic that's been lost. We know there's an organic layer to that. It's carbon rich, But that's as far as we can go. There's unfortunately no DNA. It would be great, though. Would be great.
I think we have one more.
I just had a question about the deep water ones you found. That's new to me. Why would they be living in deep water? Wouldn't it be harder for them to survive than... Wouldn't it be easy living in the shallow water?
It depends on whether you live from the energy of the sun. These organisms might have been chemosynthesizers. They could have lived from either symbiosis with bacteria that were using some high sulfur or things like in vents, sea vents. And what the quiet environments at the bottom of the ocean are doing is they're protecting you from, waves and things like that that would have basically eroded away or taken away all of the shallow water ediacarans like the ones from Flinders Ranges, which are susceptible to being washed by storms and lost. So down there it was a quiet environment, and they were probably capable of taking energy from bacteria there that were absorbing ions from the water rather than using photosynthesis as their energy provider.
One more, I think. Yep.
Given the success of the multicellular animals, why do you think it took so long for them to appear on Earth? Why did the unicellular life stage last so long?
There are several, it was a short talk, and there are several proposed triggers for the Cambrian explosion. We know from the geological record that there is an increase in a number of ions and oxygen. Oxygen surpasses a certain threshold. Organisms cannot grow, multicellular organisms cannot grow if there's not enough oxygen to diffuse from the outside to the cells in the middle. So single-cell organisms, they were capable of doing that, and even very small 1-2 millimeter organisms in the ediacaran were capable of doing that, but there was no capacity, because there wasn't enough oxygen, for them to grow into huge sizes. Why did the ediacarans do that? They might have done that, however, in symbiosis with bacteria. So how you reach that is not clear. Second, there's a number of other suggestions that might have triggered the Cambrian explosion. The other one is the Hox genes. The organisms need a number of instructions in their body to be able to define what's dorsal-ventral, what's anterior-posterior, proximal-distal. All those genes, Hox genes, appear to have started working as Hox genes in the late Ediacaran or during the Ediacaran. So basically, you don't have the instructions to build a complex organism until at least the Ediacaran. So there were a number of conditions, whether they be external, like oxygen levels, or internal, like the capacity to generate three-dimensionality with Hox genes.